I'll be talking about a piece of literature titled A Programmable Dual RNA Guided DNA Endonuclease and Adaptive Bacterial Immunity. Before I provide an introduction, I want to go over some terms that one might not necessarily know. The first is CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. The second is Cas, which simply stands for CRISPR associated. And the CRISPR-Cas system is a system that the authors were primarily interested in, which I'll explain in more detail later. Protospacers are fragments of foreign sequence, typically from invading viruses or plasmids. CrRNA stands for CRISPR RNA. Tracer RNA stands for transactivating CrRNA. And PAM stands for the protospacer adjacent motif. As I mentioned earlier, the CRISPR-Cas system is a system that the authors were primarily interested in. And the system occurs in 3 steps and provides immunity to bacteria and archaea. The first step is the adaptive phase, and this is when protospacers from the invading virus or plasmid are integrated into the host chromosome. The second step is the expression phase, and this is when the CRISPR arrays that contain the protospacers are transcribed and processed, similar in the way that DNA is transcribed into mRNA, and then the mRNA has to go through processing in order for it to become a mature RNA. Lastly, the third step is the interference phase, and this is when CrRNAs, mature CrRNAs that result from the transcription and processing, form complexes with proteins. They anneal to and cleave foreign sequences of the invading protospacers that they are complementary to. So in a way, this acts as a memory immunity in the same sense that we have memory of invaders through antibodies. We can see the steps in the image on the right, in which a virus invades the cell and the DNA is cut and integrated into a CRISPR array transcript. So the spacer is this red section here. This whole thing will be transcribed, and it'll go through processing, and eventually it'll lead to different cascades. Cleavage of viral RNA, cleavage of mRNA, and cleavage of DNA. So there are different systems of CRISPR and Cas, and these are the types of systems. There are type 1 and 3 alongside type 2. Type 1 and 3 can be grouped together. They're both found in bacteria and archaea. They both use Cas endonucleases to process the pre-CrRNAs. And they also utilize multi-Cas complexes. Whereas type 2 systems are found only in bacteria, they are the most well characterized. And instead of Cas endonucleases, RNase 3 is used to process the rRNAs. And RNase 3 can only work when it is triggered by tracer RNAs and when Cas9 is present. Overall, this piece of literature is centered around the type 2 system and the Cas9 protein. This slide shows the different types of CRISPR-Cas systems. At the very top, we can see a plasmid and we can see a linear portion of DNA. Both of them have a protospacer adjacent motif and a protospacer sequence. We can see that depending on the different types of systems, they will be processed and cut differently. For type 1, the PAM is on the left-hand side of the protospacer, whereas for type 3, the PAM is on the right-hand side for the protospacer. And in type 3, the PAM is not present. The protospacer sequence is the only thing that was cleaved. There is a Cas operon, and underneath it, downstream of it, there are various protospacer genes that have been incorporated into the array. Once transcribed, a pre-CrRNA is formed, and processing of the CrRNA will lead to a mature CrRNA. Depending on the type of system, this CrRNA will be incorporated into a Cas complex differently. For type 1, we can see that Cas3 is the hallmark protein. For type 2, Cas9 is the hallmark protein as stated earlier. And for type 3, Cas6 is one of the hallmark proteins. Cas10 is also another protein that is typically talked about when type 3 is of interest. On the left-hand side, we can also see that all of these steps are divided into the 3 respective phases, in which there's adaptation at the top, expression at the middle, and interference at the very bottom. The last portion that I'll be talking about for the introduction to this piece of literature is the research focus. The authors led into the experiments by discussing Cas9 and how Cas9 had been previously hypothesized to influence cRNA maturation and cRNA-guided DNA interference. cRNA maturation by Cas9 had been studied previously, whereas cRNA-guided DNA interference by Cas9 had not been studied. And this was a gap in the knowledge that prompted the authors to begin their experiments. On the right, I included the crystal structure of the Cas9 protein with a DNA sequence within its active site.